All right. Let's talk about the small intestine. So, leading to this point, we've talked about the previous organs, starting at the mouth, and we went down in the esophagus and stomach, and we took a little break away from those, detoured into talking about pancreas, then the liver, and the gallbladder. Okay. So we talked about pancreatic juice and the enzymes that are part of it, along with sodium bicarbonate, and then bile coming from the liver and gallbladder, all of which gets dumped into the first segment of our small intestine. Our small intestine is where most of the magic takes place. As far as magic, I mean most digestion and absorption. Okay? It's called the small intestine because it's pretty skinny in width. It's about 1 inch in diameter, but it's super long. Okay, it's about 10 feet or so. So its great length provides a large surface area available for digestion and absorption, but that is further increased by kind of 3 features. Okay, what we call circular folds that you can see here on the next page, which if you were to open up the small intestine anywhere, you can actually see with the naked eye. Then there are little finger-like extensions that extend off of the circular folds called villi. Villus is singular, okay, but there are many, many, many of them, extensions of the mucosa. We zoom in on one, we see that there are cells that have microvilli. Okay. So what this creates is total surface area of approximately 200 square meters, about the same surface area as that of a tennis court. So that's the amount of contact that our small intestine has with the, and I'm not going to call it food, I'm going to call it chyme, so that it can digest it and absorb it. Okay. A little bit more about the plicae or circular folds. They, like I said, are visible with the naked eye, 10 millimeters in length, and are folds of the mucosa and submucosa. Villi are finger-like projections of the mucosa that are 0.5 to 1 millimeter in length, extending off of those. And then microvilli are the projections of the apical or free membrane of the absorptive cells that are found on the villi and line the lumen of the small intestine. We call the microvilli of these cells, sometimes anyway, we refer to it as what we call a brush border. Okay. Now, microvilli are found on the absorptive cells, as I talked about or mentioned right here. Let's talk about the absorptive cells, which are part of the mucosal layer. Absorptive cells do exactly what they sound like. Okay, what their name implies is that they absorb, okay, and that's very aided by the microvilli that they have. But they also secrete a bunch of what we call brush border enzymes. I don't have them listed here yet, but we will get to them here in just a couple of slides, so I'll kind of wait and talk about them then. There's also goblet cells. Goblet cells are found in many places of the body. In this case, they do it just like what they do anywhere else, secrete mucus. There's also intestinal glands, which are not cells, but glands, collection of cells that secrete our intestinal juice, about 1 liter to 2 liters worth, pretty significant amount. It's a clear yellow fluid that we secrete containing water and mucus, has a slight alkaline pH of 7.6, and it aids the pancreatic juice in providing a liquid medium for absorbing substances from our chyme. Several enteroendocrine cells are found in the crevices of our intestinal glands and include our Paneth cells that secrete a bacterial enzyme, lysozyme, we've talked about many times this semester, CCK cells that secrete cholecystokinin, S cells that secrete secretin. If I go back to our slide here on the pancreas, we talked about secretin and cholecystokinin. I mentioned, make sure you know those are released by the small intestine. Now we talked about them, and then we talked about what they do for the pancreas. Okay. So those are all parts or features of the mucosa. The submucosa of the duodenum contains Brunner's glands or duodenal glands that secrete an alkaline substance mucus that neutralizes the acidic gastric acid. the gastric juice in our chyme. And so it just helps protect the small intestine from, having too much acidity, because our small intestine, like our esophagus, is not as protected from the harsh acid that our stomach is. Okay, mechanical digestion also takes place in our small intestine, includes two processes. That segmentation that I mentioned earlier in the lecture, like squeezing toothpaste from the middle of the tube. And then there's a special form of peristalsis called the migrating motility complex. Segmentation mixes chyme with digestive juices and brings the particles of food into contact with the mucosa for absorption. It does not result in the push of contents along the tract because that occurs through this migrating motility complex. This special form of peristalsis begins after most of a meal has been absorbed, and it begins in the lower portion of the stomach, pushes the chyme forward along a short stretch of small intestine before dying out. A migrating motility complex slowly migrates down the small intestine, reaching the end of the ileum in about 90 to 120 minutes before we start another one back up in the stomach. Altogether, chyme remains in our small intestine for about 3 to 5 hours. So we said in our stomach, 2 to 6 hours? Here's another 3 to 5. Once again, demonstrating that when somebody says, oh man, that meal ran right through me, 30 minutes after they ate, they are full of you-know-what. Yeah. Okay, let's talk more about the digestion of our carbs, our fats, and our proteins. So I've got a slide dedicated to each. Over on the right, we've got a figure showing you polysaccharide, which we said is typically what we should be ingesting as far as carbohydrates. Each of these little pink dots would be a monosaccharide, and a polysaccharide is many, many, many of those pink dots. And our first two enzymes that act on those polysaccharides, salivary amylase, which we know is secreted by our salivary glands, acts on those polysaccharides until it's inactivated by the acidic pH of our stomach, meaning that only a few of our starches are actually broken down by the time the chyme leaves the stomach. The other amylase enzyme is pancreatic amylase, which is released into the small intestine and continues the digestion of any remaining starches. All right, I just mentioned the term brush border enzymes. This is sucrase, lactase, and maltase that break down disaccharides, sucrose, lactose, and maltose. So, going over to our list, under carbohydrates, I'm going to add sucrase. I'm going to add maltase. And I'm going to add lactase. They break down sucrose, maltose, and lactose, respectively. Hence the name for each of them. Remember, sucrose is table sugar, where you've got glucose and fructose bonded together, so we're going to split those apart. Maltose is malted sugar, and lactose, where's lactose? Where do we get lactose from? Milk, dairy products, okay? This is the enzyme that people that have lactose intolerance have kind of faulty lactose or lactase enzyme, excuse me. So that means that they have trouble digesting dairy. So what we end up with are, hopefully anyway, singular monosaccharides, which are easily going to be absorbed through the wall of our small intestine. Now, not all carbohydrates that we eat are easily digested. Cellulose is an indigestible polysaccharide, meaning that we can't break it down. We call this fiber a lot of times, right? You've probably heard that term before. Things that are high in fiber are really good for you. What are foods that are high in fiber? Corn, absolutely good one. Any fruit or vegetable, really. There's parts Any fruit or vegetable will have lots of cellulose in it. Cellulose forms the cell wall of plants. So anything that's, made of a plant, whether it's a green bean or, ear of corn or whatever, we can't digest those parts, okay? So they come out looking just like they did when they went in, like in the form of corn, okay? Digestion of carbs ends with the production of monosaccharides, which the digestive system was able to absorb. I've already said that, so we'll move on. All right, proteins, as we know, remembering back from the beginning of the semester, we learned that proteins consist of chains of amino acids. So each one of these little green balls represents a singular amino acid. We must break them down into those at least singular or dipeptides, meaning that we have, what we prefer is one, but we can work with a couple bonded together. The first protein digesting enzyme is pepsin, released into the stomach, starts to get this ball rolling by breaking those long chains of amino acids down into shorter chains. Okay. Then we have within our pancreatic juice, trypsin that I mentioned earlier, but I've also got listed out some other ones. Chymotrypsin, carboxypeptidases, and elastase. And I listed them because they were in the figure. I'm going to not add those to the list, but I am going to add these two, aminopeptidase and dipeptidase. So going to the far right, we've got for proteins, pepsin, trypsin, and I added last time hydrochloric acid because it denatures proteins. Remember, a denatured protein is unwound protein. Unwound proteins are easier to digest. HCl is not an enzyme, okay, but it does help with digestion. So I'm going to add aminopeptidase and dipeptidase. And those are brush border enzymes. So kind of like the sucrase, lactase, maltase that we just talked about, released from the brush border by those absorptive cells of the small intestine. Like I said, we like to get them down to the point where they're individual amino acids. We can also deal with them in the form of dipeptides, meaning 2 amino acids, or even tripeptides with 3 amino acids. And then those free amino acids can be released through the intestinal epithelium and into blood capillaries before being delivered to the liver by way of the hepatic portal vein. Okay, our final of the 3 are fats to talk about. Remember, fats, the most abundant type of fat that we're ingesting is a triglyceride. Thinking back to the beginning of the semester, I'm going to actually just pull up that slide and kind of refresh your minds on what a triglyceride looks like, because I think that'll be helpful. Okay, here's what a triglyceride looks like. There's kind of 4 parts. There's 1 glycerol molecule, a 3-carbon molecule, and then there's 3 of these, what we call fatty acids. Okay, what we're going to do is just break everything down so that the fatty acids are separate from one another and the glycerols separate as well. So here you can see these kind of little yellow things. Those are each a triglyceride. You can kind of see the 3 little tails, and then the top is where the glycerol molecule is. Okay. Enzymes that split triglycerides and phospholipids, because phospholipids are another form of fat that we will get in our diet, just not nearly as plentiful. We call these lipases. Okay. We have lingual lipase that we produce in our mouth, but that doesn't begin acting until it reaches our stomach. Gastric lipase, which is produced by our stomach and starts that, or will work in our stomach as well. So those do some of the digestion, but our pancreatic lipase is our major principal fat digestion enzyme, and it's released from the pancreas into the small intestine. Okay. What we do is we break down those triglycerides into fatty acids, so the singular chains of hydrogen chlorides or hydrogen carbons, and then the monoglycerides, which is just the glycerol. Before a large lipid globule containing triglycerides can be digested in our small intestine, remember we use bile and the bile salts to take that large globule and break them down into smaller globules. We perform what's called emulsification, which is thanks to the bile produced by our liver, released by our gallbladder into the small intestine. This allows free fatty acids and monoglycerides to move into what we call a micelle, which is formed by the bile salts. We'll talk about this more here in just a couple of slides, where they can then leave the myocells, entering the intestinal epithelial cells. So we'll get into all that here in just a little bit. So looking at our list for fat, lingual lipase, gastric lipase, pancreatic lipase, those are the three principal enzymes. Bile is going to help us emulsify, and then I added phospholipase the other day because remember it's going to digest phospholipids, which are a component of our diet as far as fats go, but not nearly as important or plentiful as triglycerides. So that is our list, for fats. All right. So to summarize, 24.5 here lists off lots of enzymes. It's basically the same list as what we have here, but with a lot more that I didn't really focus on, like these from the pancreas: chymotrypsin, elastase, carboxypeptidase. I didn't tell you to worry about those. Didn't really worry about. I did mention them: ribonuclease, deoxyribonucleases. From the pancreas, and then there's a lot of those brush border enzymes that I didn't talk about. Alpha dextrinase I didn't mention, enterokinase I didn't mention. What's nice is, it's kind of my list, but it's just kind of doing it a little bit differently, telling you where the secretion is coming from, what it's breaking down, and what it's forming. So it's a little more complex as far as what I want you to know, but something you might want to study. Definitely know the list that I created. And obviously what's missing here are the non-enzyme substances like bile and hydrochloric acid that I added to our list for bile being fat and hydrochloric acid for the list of proteins. Okay, so now that we have a good idea of the digestion, let's talk absorption. The whole reason why we're eating and drinking is so that we can allow the nutrients that are in our food and beverages to enter our body. Absorption is defined as the passage of substances through the intestinal mucosa into our blood or lymph. It occurs through a variety of means: simple diffusion, facilitated diffusion, meaning it uses a protein to carry it in, osmosis, which is specific to water, and then active transport, which we know involves energy in the form of ATP. Okay, let's talk about the absorption of carbohydrates first. We can only absorb carbohydrates in the form of monosaccharides. 3 monosaccharides exist: glucose, fructose, and galactose. Okay. Other indigestible carbohydrates would be our cellulose and fibers that we leave off in our feces. So looking here at the next page, we show a picture up here at the top. We show fructose and then glucose and galactose and how they're absorbed. This is the lumen of the small intestine. Here are the absorptive cells. Here's a blood capillary. Here's a lymphatic capillary known as a lacteal, which is where the fats are going to end up going. For everything else, the carbs and the proteins, they're going into the blood. Okay. So for fructose, it's a little bit different than how we absorb glucose and galactose. Fructose, let's talk about it. Oops, wrong way. Too far. Fructose is found in fruit and is transported just by facilitated diffusion, meaning it's going to use a protein transporter. Glucose and galactose are transported via secondary active transport coupled to the active transport of sodium. Okay. Where'd that picture go? Oh, there it is. Okay, looking. So when sodium gets pulled into the cell, glucose and galactose just take a piggyback ride. And that's all that's really happening there. Fructose doesn't need a piggyback ride. It can come into the cell just fine. Once it's in the cell, then it's just going to use the same method, whether it's glucose, fructose, or galactose, leave the cell and enter our bloodstream. So carbs, nothing too crazy. Okay. Proteins, most of them are absorbed as amino acids via active transport processes. There are different transporters that carry the different types of amino acids. Some carry them similar to the way that glucose was transported, while others are actively transporting the amino acids by themselves. At least one transporter brings in dipeptides and tripeptides together with hydrogen ion. So looking at the figure, we can show, actually, I think this figure, I know it's a little bit smaller, but dipeptides and tripeptides can take a piggyback ride along with hydrogen ion. Okay. Whereas when it's a singular amino acid, it's going to be a little bit different depending on what it is. Some of them just go by themselves. Some of them have to take a piggyback ride with sodium. Okay. Now going back to the dis and the tris, when they make their way into the absorptive cell, what we have to do is hydrolyze them down to the single amino acid. So break the bonds, split them from dis to singular, 2 singles. Tris, we split them into 3 singles. Okay, so that's why we don't see any dis or tris here being moved out and then traveling into the bloodstream. We can only get them out of the absorptive cell in the form of a singular amino acid. Okay. Now, fatty acids and fats as a whole are quite a bit more difficult. Okay, so let's talk about them. All dietary lipids are absorbed, and it looks like it's kind of cut off from there, but it's via what we call simple diffusion. Thinking back to the beginning of the semester, when we talked about diffusion, we learned that simple diffusion is the easiest form of diffusion, so it's kind of going against what I was just saying. This is the most complex. Simple diffusion is possible because fats, remember, can pass through fats very easily. Plasma membrane is predominantly made up of phospholipids. Okay, but to get them to the absorptive cells, we have to form what we call micelles. Okay, now I'm getting ahead of myself here, so let me read through. As a result of the emulsification process, remember we take a big glob into smaller ones. We take our triglycerides and we break them down into monoglycerides and fatty acids. So we take our triglycerides and we break them into their different parts. The 3-carbon glycerol and the fatty acids are separated from one another. And those fatty acids can either be short chain, where there's not very many hydrogen carbons, or there can be a lot more hydrogen carbons, or long chain. Short chains are pretty super simple to get into the cell. Just go in directly by simple diffusion. The long chain fatty acids and monoglycerides need bile and bile salts to aid in them, making them more soluble so that they can pass into the absorptive cells. We form that structure I talked about earlier, what we call a micelle, which is like an Uber that carries those fat parts, the fatty acids and the monoglycerides, to the absorptive cells. Okay, then they come into the absorptive cell. The bile salts stay in the intestinal lumen. The fat comes into the absorptive cell in its pieces. So we break it apart and then we pull it in, and what we do is we put it back together, forming triglycerides again. And then we use another Uber, what we call a chylomicron, which is what this little green blob is, where they then leave by exocytosis, so bubble pops out of the cell, and unlike unlike carbs and proteins, fats go into lacteals, which are lymphatic capillaries, and these are found within the villi of the small intestine. Okay. Now what ends up happening is, remember, lymph is always going to end up being carried back to close to by the heart, where then we dump the lymph back into the bloodstream. And eventually that blood is going to carry that fat to our liver. Okay. Now with regards to amino acids and carbohydrates, remember, we have that portal vein that connects our intestines to our liver. So with carbs and proteins, we're going to carry them directly to the liver, whereas fats, we're going to get it to the liver, but it's not as direct. So the end goal is the same, but getting there is a little bit more tricky. Okay. So that's how we absorb carbs, proteins, and lipids. Those aren't the only part of our diet. A big component of our diet are electrolytes, which don't just come from our diet, but also just the stuff that we've released into the lumina of the tract. Okay. So many of the electrolytes are absorbed from GI secretions, and the rest comes from foods and liquids. Na plus is absorbed into the blood via the sodium-potassium ATPase pump after they've moved into the absorptive cells via diffusion and secondary active transport. Other ions are absorbed by active transport. I don't have a figure on either of these pages to show that, but I'm just going to tell you that's what is happening. Most of the water is absorbed in our small intestine by osmosis. A little bit of water and any remaining water that we absorb takes place in the large intestine. Water absorption here is dependent on the absorption of electrolytes and nutrients to maintain osmotic balance. Okay. We'll kind of talk about how electrolytes affect the movement of water when we get to the kidneys. But basically, water just chases any of these electrolytes, any sodium, potassium. Water goes where like salt is, right? Sodium and chloride are electrolytes. So like, if we pretended this was salt coming into the cell, water's going to go there. If salt goes out of the cell into the blood, water's going to go there. So water just is going to chase those things in order to maintain osmotic balance or water balance, essentially. Okay, vitamins, there's two types of vitamins, water soluble and fat soluble. Water soluble are absorbed just by simple diffusion. Fat soluble vitamins are absorbed just like fats are with using micelles. Now, as far as the exam goes, I'm not going to ask you to be able to explain all these different things. And I definitely would recommend that you kind of know the kind of great details about it. For example, you should know that, micelles and chylomicrons are specific to fats. We absorb carbs and proteins into the blood. Fats get absorbed into lacteals. So just kind of the major points, not the very, very specifics. I listed off the specifics, but it's not something I'm going to be very, very particular on you knowing next week. Okay, so that wraps up our discussion on the small intestine. And so far we've talked about, very recently, the pancreas, the liver, and the gallbladder that are associated with the small intestine. So that's what this table is kind of summarizing. What we have left is the very last segment of our digestive tract, our large intestine. Our large intestine is the terminal portion of the GI tract. It is a lot wider, which is where its name comes from, than our small intestine. A little over 2 inches in diameter. Length is 1.5 to 1.8 meters. What did we say that was, like 6 feet this morning, something like that? 2 key divisions of it exist. A kind of stupid dead end, cecum, and then the rest is the colon. There's an ascending, a transverse, a descending, a sigmoid, colon, and then the rectum is the last segment that leads out to the anus. Overall, what our functions of the large intestine is to complete the absorption, mainly of water, production of certain vitamins, formation of feces, and then the expulsion of that feces from the body. Within our large intestine, we have a large population of bacteria that we call our intestinal microbiota. That provides us, their host, with several benefits. So we provide them with the food that we're eating. They get some of that. And then they provide us with some benefits. So it's a mutualistic relationship. So both parties are going to be benefiting from this relationship. They produce vitamin K and folic acid, which are absorbed across the wall of our large intestine. They produce short-chain fatty acids, which can be used for energy by cells of the colon and can aid in the absorption of different materials. And they reduce the ability of any pathogenic bacteria causing disease. Anything that means pathogenic means it causes disease by promoting anti-inflammatory processes. Final stages of digestion occur in our colon. And there's two different ways that it's done, mechanical and chemical means. Mechanical and digestion involves haustral churning. I talked a little bit about this in anatomy this morning. But if I go back to this picture of the large intestine, you can see these like pouch-like features. These are called haustra. And what we do in a haustra is we churn that chyme and then we pass it to the next. And then it churns and passes to the next and passes and passes and passes, churns and churns and churns. And so that's a great way to continue to digest and absorb. Peristalsis also occurs in the large intestine, but at a slower rate than in the more proximal portions of our tract. A final type of movement is called mass movement, a strong wave of contraction beginning at about the middle of the transverse colon, quickly driving the contents of the colon into the rectum. It's similar to peristalsis, except that the contraction lasts for a longer period of time. Usually takes place about 3 to 4 times a day during or immediately after a meal. The presence of food in our stomach triggers this to occur in our large intestine. This is why people have that urge to go to the bathroom like right after a meal. Not because that meal ran right through them. It's because this chyme, or you could probably call it at this point feces, in their large intestine was sitting still, but then the stomach turned on mass movement, and now everything's going down the line. And the rectum's starting to fill up, and now that person has the urge to go. Okay? We call this a gastrocolic reflex. Gastro for gastric, stomach, colic for colon. Okay? Chemical digestion involves mucus secretion by the glands of the large intestine. We do not produce any enzymes. So our list that I've been keeping, the tally, is finished. We do not need to add anything to it. And the chyme here is prepped for elimination by the action of bacteria by fermenting any remaining carbs. We'll talk about fermentation in our next lecture. But this is what's going to cause methane to be produced, which is what contributes to flatulence. Flatulence, or what we call farts more often than not. So by using our bacteria and digesting the remaining carbs, this is why we have gas. Okay. Bacteria also aids in breaking down any remaining proteins into amino acids, as well as the decomposition of bilirubin, giving feces that typical brown color, as I was talking about earlier in the lecture. Any remaining water not absorbed by the small intestine should be absorbed by the large intestine, and along with it will come electrolytes like sodium and potassium and chloride and stuff like that. So just to kind of reiterate where the absorption takes place, I really like this figure. It's kind of a weird looking figure, but it does a job of explaining where stuff is absorbed. Majority of everything is absorbed in our small intestine, a little bit in our stomach, very little in our colon. No food really absorbed here. By the time we're in the rectum, nothing's absorbed, because at this point it's feces ready to be eliminated. We can talk about that now. Elimination is described as the expulsion of feces from our digestive tract, act that we call defecation. The mass movements that I talked about just a second ago push the fecal material from our sigmoid colon into the rectum. Sigmoid colon was this kind of S-shaped part of the colon. So we're going to push it down into this downward running rectum. Okay. This is going to cause the rectal wall to stretch. So when the rectum's empty, the rectum is kind of collapsed. But when the rectum fills up, it stretches or distends, which activates stretch receptors in the wall of the reflex to initiate what we call a defecation reflex. It's ultimately going to empty the rectum. It occurs as follows. In response to distension of the rectal wall, receptors send sensory input to the sacral spinal cord. So to the tail of our spinal cord. Output from motor neurons in the cord travel along parasympathetic nerves back to the descending sigmoid colon, the rectum, and the anus. So we're going to send signals from our spinal cord to a good portion of our rectum, anus, colon, all the way, throughout the large intestine, essentially. Resulting contraction of the longitudinal rectal muscles will shorten the rectum, which increases the pressure inside of it. So we're kind of pushing stuff, but we're not opening up the anus at this point. This pressure, along with the parasympathetic stimulation, opens what we call an internal anal sphincter, which was contracted when the rectum was empty, but now is relaxed when the rectum is full. Okay. Now your internal anal sphincter is under involuntary control. We cannot control that with our own conscious awareness, and that's because it's made up of smooth muscle. Your external anal sphincter is voluntarily controlled. It's made up of skeletal muscle. Okay. What happens is when our rectum fills up, it contracts, whereas when the rectum was empty, it's relaxed. So we flip these. External's relaxed, internal's contracted. Now the external's contracted, the internal's relaxed. If we voluntarily relax it, so we purposely relax it, the anal canal opens up and defecation occurs. We expel the feces and we push that feces out. This is also assisted by what we call a Valsalva maneuver, involving voluntary contractions of our ab muscles and a forced exhalation of a closed larynx. Okay. We do this all the time. We don't really realize it. Okay. But like when a person lifts something really heavy, they do this a lot of times. It's like, holding your breath, not blowing out, but blowing internally. Okay. Kind of a weird way to describe it, but that's what you're doing. Okay. Next time you go to the bathroom, you can think about what you're doing. You don't have to do it right now. This is going to increase pressure within the abdomen, pushing the walls of the sigmoid colon and rectum inward. A person can postpone defecation by voluntarily contracting that external anal sphincter. If they just hold that, they're not going to go to the bathroom. The feces eventually backs up into the sigmoid colon into the next mass movement, stimulates the stretch receptors, again creating the urge to defecate. That's kind of why you get that urge, and then if you don't go, it might disappear and then come back, and then it might disappear if you don't go again. In infants, defecation reflex causes automatic emptying of the rectum because they have not gained voluntary control over the external anal sphincter. So this is why babies, it takes years for them to get control and then get out of diapers. Something I would love to get my 2-year-old out of, but it's still pretty early. Constipation, we all know what that is, but that's where the contents of the lower part of the colon and rectum move at a slower rate than normal, which means that extra water is absorbed from the feces and the stool is hardened as a result. Diarrhea, we all know what that is, but this is where the speed in which we are pushing the stuff along, the motility is too high in the small intestine, and that causes a decreased absorption of water and electrolytes, and that's why it's a watery stool. Okay, so now we've covered the entirety of the digestive tract. Really quickly, I want to talk about the digestive phases or phases of digestion. There are 3 overlapping phases, meaning that they aren't taking place independently. They're taking place at the same time because you've got food in different places within the tract. The first is the cephalic phase, which refers to regulation by the brain. Cephalic means head, so that's where this is, being initiated. It involves digestion even before we may have even eaten. Okay. The smell, the sight, the thought, or initial taste of food activates centers within our cerebral cortex, hypothalamus, and our brainstem, which then results in activation of 3 different pairs of cranial nerves: facial, glossopharyngeal, and vagus. The facial and glossopharyngeal nerves stimulate the salivary glands to start secreting saliva. This is why, like your dog, our dog does this all the time when we're eating dinner, just drools all over the floor. She's drooling because she sees and smells the food. She's not eating until I toss her some stuff, but she's producing a lot of saliva. What we can't see is what's actually taking place in her stomach as well. The vagus nerve is going to stimulate those gastric glands in the stomach to secrete gastric juice. I always think of this like stretching before you go for a jog. Okay, it's getting the digestive system ready for the arrival of food that's about to be eaten. Okay. Once again, gastric is a term that refers to stomach. This gastric phase occurs when food arrives in the stomach and is controlled via neural and hormonal mechanisms. I kind of talked a little bit about it earlier in the lecture. Neuroregulation is stimulated when the food in the stomach arrives and activates the stretch receptors as our stomach expands. There's also those chemoreceptors in the stomach that monitor pH change due to the presence of proteins in the stomach. These changes, so the stretch and the change in pH, stimulate the parasympathetic and enteric neurons, resulting in waves of peristalsis and the flow of gastric juice from the gastric glands. So when your stomach starts to fill and when its pH changes, you start contracting the muscles to start digestion mechanically, and the release of the juices to start digestion chemically. Hormonal regulation is regulated by that hormone we mentioned earlier, gastrin, released in response to distension of the stomach, partially digested proteins in chyme, high pH of chyme due to food in the stomach, caffeine in chyme, and acetylcholine released from our parasympathetic nervous system. Gastrin stimulates large secretion of gastric juice, strengthens contraction of LES, lower esophageal sphincter, preventing reflux, increases motility of the stomach, and relaxes the pyloric sphincter to promote gastric emptying. Now, these first 2 phases are going to enhance digestion. They're really getting the ball rolling, getting things going. The intestinal phase kind of does the opposite, and it'll make sense after I describe why we do this. We call it the intestinal phase because this is what is activated when that, it says food, but we could call it chyme at this point, enters the small intestine. In contrast to reflexes initiated during the cephalic and gastric phases, stimulating the stomach to secrete and to be motile, those occurring during this phase have inhibitory effects that slow the exit of chyme from the stomach, okay, which is a preventative protective mechanism from the duodenum being overloaded with more chyme than it can handle. So basically, it's going to make the stomach empty really slowly. Okay. So, in addition, responses occurring during the intestinal phase promote continued digestion of foods that have reached the small intestine. Arrival of chyme in the duodenum stimulates sensory neurons, evoking a neural reflex, inhibiting gastric motility and secretion. So as we start dumping stuff out of the stomach into the small intestine sends a signal back to the stomach that says, slow down. That keeps the stuff in the stomach for a longer period of time, and it gives the small intestine time to do the digesting it needs to do, especially if you're eating anything fatty. Presence of fat in the chyme is really difficult to digest, so we need to slow the emptying of fat from the stomach. So the fat stays in the stomach for longer periods of time, allowing the intestine more time to process that fatty chyme. Does that make sense? Okay, any questions? How do you produce bile? Well, your gallbladder doesn't produce the bile, it just stores your bile. So you're going to be fine without a gallbladder. You just won't have as much bile to help digest the fat that you are eating. So, you know, it might be something you might want to manage what you eat. But a lot of people have their gallbladder out and survive just fine. Okay, well, then we can move on.